Hey, how's it going, everyone? This is FixerMed. Welcome to my high yield genetics review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 1 of my multi part video series covering the entire discipline of genetics in a comprehensive but high yield manner. So, without further ado, let's go ahead and get started on today's lesson. So, I'd like to open up by talking about the law of segregation. The law of segregation states that each individual has two alleles for each gene, one that's inherited from each parent, and these alleles segregate during the formation of gametes. Mendel's experiments, Mendel's famous pea plant experiments, demonstrated the separation of alleles during the formation of gametes. And for an example, let's consider a monohybrid cross where a homozygous dominant individual is crossed with a homozygous recessive individual. That would result in a 1-to-1 ratio of the resulting genotypes in the F1 generation. So all the genotypes would be big R, little r. And I have a cross here. It is between two heterozygous individuals, so it shows a big R, little r. And as you can see in this case, 3 of them would have the dominant gene, and 1 of them would have the recessive gene completely, while 2 of them would be heterozygous for the dominant and recessive gene. So let's go ahead and move on. Next, we have the law of independent assortment. The law of independent assortment states that alleles for different traits segregate independently during the formation of gametes. Mendel's experiments, once again, Mendel performed dihybrid crosses as well to observe the inheritance of 2 different traits. For example, crossing plants differing in both flower color, big Y, or little y, and seed texture, big S, or little s, would result in a 9-to-3 to 3-to-1 ratio of phenotypes in the F2 generation. Moving on. So, let's talk about the types of inheritance patterns. We have the dominant inheritance pattern. The dominant inheritance occurs when only one copy of the dominant allele is needed for the expression of the trait. Dominant traits typically mask the expression of recessive alleles. Examples include brown eyes over blue eyes, or curly hair over straight hair. Next, we have recessive inheritance. Recessive inheritance requires 2 copies of the recessive allele for the expression of the trait. Recessive traits are only expressed in the absence of a dominant allele. Examples include blue eyes when both alleles are recessive, or Tay-Sachs disease when both parents are carriers. Blue eyes can be characterized by little b, Tay-Sachs characterized by little r. Moving on. Codominance. Codominance occurs when both alleles contribute to the phenotype in a heterozygous individual. Both alleles are fully expressed without blending. Examples include blood type, big A, big B, where both A and B antigens are expressed on the surface of red blood cells, or a roan coat color in cattle, where the red and white hairs coexist. Incomplete dominance. In incomplete dominance, the heterozygous phenotype is an intermediate blend of the 2 homozygous phenotypes. Examples include pink flowers, RW, resulting from the blending of red, big R, and white, big W, flower colors. Sickle cell trait, HBAS, without intermediate symptoms compared to homozygous sickle cell disease, HBSS. So this shows that these individuals with sickle cell, that are heterozygous for the sickle cell gene, have less severe symptoms when compared to the individuals that have both alleles required for the sickle cell disease. Moving on. Next, we're going to be talking about pedigrees. Pedigree analysis involves the study of family trees to understand the inheritance of genetic traits across generations. It helps identify patterns, infer genotypes, and predict the risk of genetic disorders within the family. Symbols and conventions used are to commonly explain that squares represent males, circles represent females, shaded represents affected individuals, etc. Key terminologies include carriers affected, unaffected consanguinity, and generations. So carriers may have the recessive allele, but they're not affected with the disease, so they can be like big S, little s. So they carry that recessive allele, but they're not plagued with symptoms related to the disease. Affected individuals, they might be recessive for a disease, or in cases like autosomal dominant diseases like Huntington's disease, they might be affected even if they have the dominant allele. So keep that in mind. Unaffected individuals don't have the recessive allele. Consanguinity refers to interfamily relations, mainly marriage between cousins resulting in offspring, etc. And generations typically just refers to like great grandparents, parents, the current generation, children of that generation, so on and so forth. It's just to identify and mark which generation is affected with a disease to track it better. Moving on. I kind of gave some hints about autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive earlier, so let's go ahead and talk about these in greater detail. Autosomal dominant. These cannot be recessive, as two affected parents could not have an unaffected offspring, so the parents had to be heterozygous. Okay, you commonly see in autosomal dominant diseases that if an individual is affected, they don't have the two affected alleles. They're most likely carriers if it is autosomal dominant, since if you have the two affected alleles for the autosomal dominant diseases, that results in death usually. Moving on. Next, we have autosomal recessive. It cannot be a dominant here, as two unaffected parents cannot have an affected offspring. The parents must be heterozygous. So this could be like the big R, little r that I was talking about in the first slide. Both of the parents are not affected, but they had to be carriers for one of their children to be affected here. Moving on. We have X-linked dominant. Sex linkage cannot be confirmed. 100% incidence of affected daughters from an affected father suggests X-linked dominance. Basically, you see, you can only get the X or Y chromosome from your father. So if the X chromosome is affected in a male individual, then his daughters should all get the disease if it's an X-linked dominant. Meanwhile, you can see that the sons of the affected individual in generation 3, none of them got the disease, but he got the disease from his mother because the mom passes down the X chromosome, the dad passed down the Y chromosome. It's all on the X chromosome, so all the sons who got the X chromosome from their mom are fine because the dad gives the Y chromosome to them, Y chromosome, but the dad gives the X chromosome to all the daughters, and that shows that this would be an example of X-linked dominance. Let's go ahead and see what X-linked recessive inheritance looks like. Sex linkage cannot be confirmed. 100% incidence of affected son from an affected mother suggests X-linked recessive. As you can see, similar premise. Mom passes down the X chromosome to the daughter, and then dad passes down another X chromosome to the daughter. Since the daughter needs both X chromosomes, she could be affected with an X-linked recessive disease, and then she marries someone who does not have any affected X alleles and has a Y allele. So he passes down the clean X chromosome to all the daughters, but the mom passes down the affected X chromosome to all the sons. Therefore, this would be an example of X-linked recessive inheritance. Now, mitochondrial inheritance, this basically refers to the mom passing down the disease to all her offspring. As you can see, in the first generation, mom is affected. In the second generation, all of her kids are affected. And then the moms in the second generation, they all pass it down to their kids as well. But the son doesn't pass it down, since the mitochondrial genes only come from the mother. That's a good way to remember that. Very easy to spot compared to some of these. It takes some practice getting used to the pedigrees, but Genetics is my favorite unit, so if you guys have any questions about pedigrees, feel free to leave them in the comment section below. Now, let's go ahead and move on to some questions. A couple, both carriers of a recessive genetic disorder, is planning to start a family. The disorder manifests only in individuals with 2 copies of the mutant allele. What is the probability that their first child will be affected by the disorder? Pro tip, draw your Punnett square out. It should help you out a lot. And as you can see, they are carriers of a recessive genetic disorder. So, hint, hint. All right, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. If not, moving on now. So since both parents are carriers, that means they both carry 1 normal allele and 1 mutant allele for the recessive disorder. The possible combinations for their offspring are big N, which means they're completely normal, big N, little n, which means they're a carrier, or little n, which means they're affected. Therefore, the probability of having an affected child, little n, is 1 out of 4, or 25%. This is due to the Mendelian principle of autosomal recessive inheritance, where 2 copies of the mutant allele are necessary for the expression of the disorder. Basically, draw your Punnett square out. They're both carriers, so big N. The only possible alleles are 1 in 4 chance of getting a big N, 2 in 4 chance of getting a big N, little n, or a 1 in 4 chance of getting a little n. So only a 1 in 4 chance of getting a offspring that's actually affected. And you guys can read this if you want, but I think the explanations are not the best here, so I'm just going to move on to the next question. I think the way I explain it with the Punnett square is much better. Next, we have a couple that has a child with a rare autosomal dominant disorder. The father is affected and the mother is unaffected. What is the probability that their next child will also be affected? Remember what I said about autosomal dominant disorders. Patients with autosomal dominant disorders typically don't have 2 copies of the same allele that causes the disorder. All right, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. If you did the Punnett square, you should have gotten this answer right. In autosomal dominant disorders, a child has a 50% chance of inheriting the dominant allele from an affected parent. Since the father is affected, heterozygous big A, little a, there are 2 possible combinations for his genotype, big A, or big A, little a. Therefore, the probability of passing the dominant allele to the next child is 50%. The mother's genotype is irrelevant in this case, as the disorder is dominant, and having one copy of the dominant allele is sufficient for expression. Therefore, the correct answer is 50%. See explanations for why the other answer choices are incorrect are here. Yeah, guys. If you have any questions, comments, or concerns, please feel free to leave them in the comment section. If not, this is FixerMed signing off. Hope you guys have a great day and hope you enjoy this new review series for genetics. All right, good luck studying and goodbye, everyone.